name is Darren Chang, and today I'll be showing you how to find clonally related cells and identify additional mutations as well as determine whether or not the cells are clonally related in situ or culture artifacts. This step can best be done with two cells that are related to one another. However, this can be done with 3 or more related clones by doing this repeatedly over unique pairs of clones. So first, you have created a master mutation list. You can do this by first running the master mutation list creation script on your mml.xl. This creates a named Excel file which you can then open up. First thing you want to do is to make sure that there are no manually inspect calls, which there aren't. Secondly, what you want to do is take every single mutation with above a 15% mutant allele frequency, copy the column K in the Excel, paste that into the master mutation list over here, and remember the name, write back 11, donor 38, and copy that down for every mutation here. What you then want to do is take all of the mutations, sort them by column A, which orders them by their mutations, chromosomal and positional. And what you want to do is create an if statement where if the A2 is equal to A1, 1, and 0. This has it so if the mutation below is shared with the mutation above it, then print a 1. If not, print a 0. So you want to drag this all the way down to the bottom of your mutation list. And then in column C, you want to search for 1. You want to look for mutations that are shared between cells. So as we go along, we find that writeback 8 and writeback 11 have 2 shared mutations. That has a pretty good chance of those 2 being clonally related. We find it again. And so at this point, we have built enough confidence to say that those 2 are related. This mutation here, shared between writeback 2, 4, and 5, upon manual inspection, is found to actually be a, because there's only 1 mutation shared between these 3, and it's this one, you'll actually find that when comparing these 3 samples to the bulk, that the mutation itself is actually an alignment artifact. So you have to watch out for these. Do not just immediately assume that because there's a mutation that is shared, it could very easily be a sequencing artifact. So there's no guarantee that these are clonally related. However, we've built confidence with writeback 8 and 11 that these 2 are clonally related somehow. So since we have some confidence, what we need to do is first run the also in script. You can do that by drag and dropping the also in script under sequencing data, master mutation list scripts. Let's save this. And drag and dropping the master mutation list XLs, the named ones, for both of the clonally related samples, writeback 8 and writeback 11. Hit enter. We'll just have to wait a moment. This will create 2 text outputs. Writeback 11 not in writeback 8, and another with an mpileUp text. We want to use the mpileUp text output as our input for the next script, as sometimes there are DNPs, or dual nucleotide polymorphisms, which MPileup cannot handle. MPileup can only handle single nucleotide polymorphisms. So my script translates the first of the two DNP points, base pairs. So what you want to do is then grab the also in MPileup script, and you'll be selecting one of these two depending on whether the sample is UCSF 500 or DNA or exome. In our sample, we only have DNA available. So first we want to drag the DNA script, the MPileup text, the DNA of that sample, the realigned, recalibrated BAM, and the star genome RNA BAM. We want to do the exact same for the other sample, the other related sample. MPileup, DNA BAM, star genome RNA BAM. This can take a couple seconds to a few hours depending on how many mutations are not related to one another. Seems like this is pretty short. So what we need to do now is open up the non-MPileup text file. That is our output from our MPileup, as well as all the maths are correct. All we need to do is double check to see if the allele frequencies are high enough to call them clonally related. With this sample, they're both around 50%, so these are good to add into the master mutation list. So you just want to open the master mutation list, copy and paste, save that. And do it again for the other sample. Make sure if there's a math that is too low, to either, that is very low, either a math of maybe around 10% or 15% or below, to probably not include that in the, when bringing all the mutations back into the original master mutation list. However, for all these samples, they seem okay, so we can add them back. Make sure that the columns are all aligned, which it looks like they are. Save that. So what we're doing here is looking at all the mutations that were not shared between the two master mutation lists and comparing them. So we found that actually there were a few mutations that initially were not found in the other master mutation list. However, when redoing MPileup, we found that they actually are. So we need to go back. We want to redo the also in R script. You want to drag and drop the same master mutation list files. We can open these up just to make sure that they're okay. This looks good. So then what we need to do is run the validation script. You want to do this with both samples. So once that's completed, you want to again look at the master mutation list just to double check that everything is okay. Look for any manually inspects. In this sample, there doesn't seem to be any, so we just need to double check with the other. Seems good. So we then want to run the final rock curve script, which we'll just drag and drop again each file. And apply the rock curve script to both samples. To finalize, just convert everything to numbers. Make sure again at the very end there's no clonally related, or not clonally related, manually inspects. And yeah, this is it. So save that. And so based off of the information we see here, there's actually a very high chance that these two samples are probably cultural artifacts, because out of the 12 mutations found, all of them are related to one another. However, there's no guarantee with that few number of mutations. There might be more confidence if there was, say, 50 cell or 50 mutations that were all related to one another. But in this particular sample, these are actually two keratinocytes that are related. And keratinocytes, at least currently in our single cell clonal expansions, tend to grow out pretty quickly, so it's pretty likely that these are cultural artifacts. However, we just need to finalize all this. It looks all good, and that is it. Again, if you want to do this with 3 or more samples, you just need to apply this method several times over unique samples. So for example, if writeback 10 was also related, you would need to compare 8 and 10, 8 and 11, and 10 and 11. And you would repeat this with additional samples if they were actually related.